don't know me, I I record these, I convert them to YouTubes, I embed them and send them to you, or embed them with Blackboard. And for those that want to listen to them, that's fine. I suggest that you do. I mean, I know how many people are listening out of the people I send it to. Most of the time, you're just listening to my review recordings, but that's okay. It's your call, not my call. Welcome to Micro. So, and it's always a little challenging because I have to wait till Dr. Elfin, who's the chair of math, finishes with her class in here. But she's sensitive to it, so it makes it a little bit easier. So I may have to get in here first thing in the morning and load some stuff in preparation for us being here. But all that aside, this is a very I most of you have me for AMP. And the way I framed it to the other classes I have at the 9 o'clock classes, now this is another step up for most of you. You're taking 2 science classes with laboratories. Okay, and so everything has elevated in the intensity and the amount of time that is going to be involved in this. And where AMP1 was a challenge in a lot of ways, AMP2 is very much about the physiology of the body. So you're really applying all of what basically bones, muscles, nerves, what skin, all the tissues and stuff like that. You're applying that. So there's a lot this is where you're going to understand how the body works. On top of that, you're stuck with an entirely new feature here called microbiology. And the most difficult part about this is our focus point. The vast majority of the course involves 2 kinds of microbes that cause problems and we have to coexist with on a relatively friendly basis. And those are bacteria and viruses. And they're totally different. We spent all of AP1 talking about what's called the eukaryotic cell, the nucleus, the mitochondria, smooth and rough endoplasmic reticulum. I will drone on. You heard me. Drone on. Don't smile back there because you heard the drone. You heard me talk about that. This is all different. This is an entirely different cell. It's called a prokaryotic cell. It doesn't have a nucleus. It doesn't have any membrane-bound organelles. It's totally different. It's constructed differently. It's much less complicated, and that's where it's so important to understand. And so that's where bacteria come from. And then we're not done yet. We're going to deal with something that isn't even alive, called a virus. I know I had a live virus vaccine. Bull. I was going to say a bad word. No, you did not, because viruses they're not living. They're not made out of cells. Remember that whole cell theory thing? All living things are made out of cells, right? Not viruses. Totally foreign. Pasteur coined the term virus before he knew what they were, because he knew there was something in the air that was causing significant problems that were there. Okay, so he knew about that. Actually, it was all about rabies virus when we start looking at historical figures, because some aspect of that word in French meant poison. And effectively, that's a lot of what viruses are, kind of replicating poisons that target various cells and tissues and create damage. So they're totally different. It is an entirely foreign subject that we're going to be talking about that's there. And yes, on top of that, there still are cells that are eukaryotic cells that are invisible to the naked eye that we call microbes. And believe it or not, they're just as big of a headache, more so. They're harder to treat because when we use antibiotics and antivirals, okay, we can target things that we don't have. When we target cells like we call protozoans, and I'll explain that in a second, we use those medications are targeting cells that are similar to ours, ergo, more side effects. Because they're going to target mechanisms that we have in our own cells. It's very hard to exclude them. We're going to get into all of that. That just gives you a level of complexity. Just starting. So this is a really This and the vast majority of the students here are in pre-healthcare careers, typically pre-nursing, pre-health. What's that one? Yeah, the one that's over in the other building. That's not nursing. Health what? Some of you are in that major. I give up. The ones that don't have to get a B in the course. Nevertheless, we do have some folks here. This is a different micro than the bio majors take. Bio majors take one that's geared more toward the scientific aspects of microbiology, the molecular biology aspects, special ways that things replicate and controls, things that we won't cover, something called the lac operon, which you don't want to know about, but it's taught in classical microbiology. Because of my background, which for those who do not know is clinical, I treated patients for 25 years. I operated on people for 25 years. I know my way around infections. Nothing recently, but the principles are still the same. But my focus is clinical, so I spend the last third of this class basically on diseases. Some of which I'm entirely too familiar with, and some of which I am familiar with because of my background, etc. So that's the starting point. Let me show you the syllabus. I got to get used to where all these things are and Blackboard and stuff. So if you go over to your Blackboard thing, and I have it now as a file out there, this would be under the Start Here tab, and you'll see where the syllabus, and you click down, you'll see you can't miss it. This is really annoying in the way. That thing, whatever that is. So here it is, very straightforward. Okay, for those that didn't have me, for those that have me, same thing. We don't do extra credit. We take 4 exams, typically. The last of the 4 is the final. It is not cumulative. You have enough to deal with. Announced exams, study guide, in-class exams, da. There is no such thing as extra credit. Don't want to hear it. Okay, you're getting to the point where, and the reason, let me frame it differently. For those that weren't here last term was an odd term for a variety of reasons. For those who know, I made a concerted effort to get a lot of people through who were having difficulty, because I felt that there was a burden put on you because of what happened with the A&P lab and a lot of other factors that were there. To that extent, I mean, when you have an open book final, you're not going to have one this time. It's just not going to happen. Don't get to that point. I want you to understand, I'm not doing it to be mean or to be. You have to grow with this because of where you're going. If you get jumped through to the next level without that appropriate preparation, you're going to bomb. And that's not helping you. Those whole student loan things are nasty. They come back to bite you. I'm being very honest with you. I'm talking to you the way a parent, a grandparent, a great-grandparent, you got the whole drill. Like that. Oh, shout-out day. My wife and my ex-wife were both born in January. My wife's birthday was the 9th. It's a few years. She's 7 years younger. My ex, we're all very, very close. We're going to have a shared birthday party over the weekend. She's at the 3/4 pole. 75 today. God bless. Doing okay. And yours truly will hit that same mark on May 10th. So keep big presents in mind. For those that don't know me, I'm always trying to get presents. Insufferably. Nevertheless, that's what I'm trying to tell you. You're going to. There was kind of a pass by intent. I do it for a lot of reasons. I have found it increasingly difficult since the COVID, and we do a whole unit on COVID, and I'll acquaint you with some of the misconceptions people have about it and the misinformation and a lot of things that they really didn't tell you and still don't about a lot of these things in the vaccine crazy world that we live in. I'm saying that honestly. It changed because we went to so much remote stuff, and it changed particularly for you folks. I mean, it altered a lot of the experience that you had preparing for, for undergraduate levels, for going to colleges and universities. And I mean, I think there was a distinct lack of preparation for many people that it impacted. So I need to. I mean, I was by intent trying to grade more reasonably. I was trying to provide alternative ways, and plus, I mean, there was a lot going on. Was there? So that maybe I'll frame it like that before I start with my talk. I only want to take another minute. I'm not going to read the syllabus to you. You can read it. 4 exams. The textbook is on there. I understand people did not have any problem logging in to the textbook. I have the McGraw-Hill people do it for me anyway. I don't even want to play with that. Okay, so that's what it is. Heck, if you don't want to have a textbook, if you're not into that, that's fine. You can find. I mean, you can find. Micro hasn't changed that much. You can get a third edition of this Cowan textbook, for probably a used one for probably $10, $15 on Amazon hard copy, hardback copy. Easy. If you like that, that's fine. I'm not here to sell you books. They're there, blah, 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 attendance, exam-wise here. You'll see on Blackboard. When I loaded this up, so it's all under Unit 1, you can see, and I have the outline following this, and I'll show you that in a second. I have the chapters. Chapter 2 is lab. I put that in there because I don't know if Dr. Dress or whoever else is doing lab are showing you, or they may have their own PowerPoints. I think doesn't Dr. Dress, I think some people had them. I think he has his own PowerPoints that he creates from that. He's nice, I'm not. And so, but I mean, I thought this would be helpful. Note that we'll be looking at chapter 1. We're going to start with a lot of, I love the introductory stuff. 3 and 4 is more about the basic bacteria, the basic anatomy. 6 is more about the basic physiology of these microbes. So, and I don't know exactly how much of that will be on the first exam. If optimally, I would like to get the relevant material out of 6, which is how microorganisms grow and their basic physiology. That's my opt for the first exam. We'll probably be at least 4 weeks in before we get there, I would think. This is an outline. I have the outline here. For instance, a general outline I have from years ago, and I still continue to use it. I look at the historical figures. I have some other handouts about the historical figures, some other handouts of some of the material I'm going to present today that you'll see subsequently. Talking about the role that microorganisms play, some medical aspects, really important stuff about emerging and resurgent diseases, very, very topical today. They're there, a lot of stuff. So you can, so I hope this will be helpful for you. So, I mean, this is just the material we're going to cover to start. That's there, just a quick snapshot, that historical figures. I do a lot about that. So we begin to look at where about the start of vaccines came a long time ago. That was how the development of smallpox. And these are all how we got to microbiology today. Very, very interesting. So that's the starting point. I took an extra minute from this morning at 9. I'm losing my touch. Voila. Yeah, I love the textbook. I have a lot of there's another PowerPoint sequence I will be interjecting and using because I like their graphics. They're both McGraw-Hill, so they're very, very similar. I have taught with this Cowan textbook off and on for 15 years at different institutions. We switched to it here from the textbook that I'll be grabbing stuff from because I'm used to it, and it'll be called Nestor. And I will have, because I like some of the illustrations, I like some of the animations better. So there's a lot of good stuff in both that we will be employing as well. And I'll frame it for you, and you'll see it. And I'll make sure you're aware of it because I'll point out to it in lecture where to find these things. They're all there. I want to give you every possible resource. I'll try to get the study guide posted very early. because I don't know exactly where it ends, but at least I can give you what I know will be on there. And maybe 2 weeks from now, I'll know a little bit more about what will be on there, and then we'll figure all that, the mechanics of that out. We typically will have exams the same day with the 9 o'clock class and the 11 o'clock class. Voice. Few of you asked me, I forget, how was the break? And I said my wife had the flu, and it really impacted it. I mean, I had about 4 days. Maybe mine was milder. Maybe I had just a virus that came on right about. I think I hammered around Christmas, and I was in big trouble. And but it disappeared. My wife was down for 2 weeks. That's part of it. So, for instance, I did it when I decided to do a little research. I mean, we haven't gotten flu shots for the last few years. Their efficiency has gone down because viruses are totally unlike us genetically. They mutate a lot. We're going to learn about viruses. Influenza is an RNA type virus, an RNA virus. RNA viruses live to mutate. COVID is an RNA virus. All they do is mutate. They're inherently genetically unstable as they replicate. And then statistically, that flu shot this year, they're looking at maybe under 40% effective. I do not know that offsets, depending on how healthy you are, having that immunization, if you choose to. She got clobbered for 2 weeks. That was an injury, and that wasn't swine flu or bird flu that we've heard about. That was influenza type A, and we'll learn about the character. It was called H3N2, and we'll learn about what that means to understand and why it's genetically unstable. Just for context, because somebody asked me something about that. The biggest problem we face today is this genetic instability, and what troubles us so much in the microbial world and dealing with it is because things are constantly changing. For instance, most of you have heard about a condition or a disease called MRSA, correct? MRSA stands for methicillin-resistant Staphylococcus aureus. The biggest single problem we face with regard to bacteria and the treatment of those that cause disease, and not a great many of them cause disease, surprisingly, only a couple of thousand, is that we are running out of antibiotics. When penicillin was identified, it was 90% effective against Staphylococcus aureus. Staphylococcus aureus is the most common infecting organism that we feed. It's on our skin all the time. We grow it in our nose all the time. So when you hear about someone with a staph infection, that's what we're talking about. Through injudicious, for inappropriate use of antibiotics, which has continued for generations, we have evolved this species twice in a span of, since the development of penicillin, which, let's say, when it became more common in use was about 1941. I'll tell you the background when we do our antibiotic unit when we get there. And so in the 85 years approximately, first, after about 40 of those years, we saw the development of what were called penicillin-resistant Staphylococcus. And so we had to modify it chemically. And we came up with different kinds. We came up with amoxicillin, and we came up with something called methicillin to specifically fight that. And what happened after another 30 to 40 years, all of a sudden, now we have developed, we bombarded it with so much of it, now we have a thing called MRSA, methicillin-resistant Staphylococcus aureus. Two different kinds, two different general kinds, one from the hospital, one in the community. The hospital one is unbelievably difficult to treat. So we have changed it now from 90% effective when we've used penicillin. Now we have 90% that are resistant. And now even to the medication we changed to combat that resistance, now it's about 70% of the strains are resistant to that. We've got to find better ways to treat these things. We're running out. So mutations, we're going to go into the genetics just to give you some context about what's going on with regard to. So We're going to learn about these, and colonize isn't bad. Maybe you'll do it in the lab. 100% of your skin, 100% of the time, will have staphylococcus on it. It will. You all look relatively neat and clean. I'll bet some of you showered this morning. There is still staph on there. We are completely covered with that. You're covered with yeasts. You're an alien, came from outer space alien, came down and looked at us and could look at our skin. They would see teeming. My God, it's a big bag of water that's covered in microorganisms. What we look like. They are your friends. Okay? They are normally there and they're supposed to be there. They're only your friends, not your friends, when something inside of you prevents you from fighting them off if they do end up getting inside of you. We'll go into that. Okay? So colonize isn't necessarily a bad term. It depends where. We're going to look at the fact that we're happily breathing air has a lot to do with microorganisms. The vast majority of the oxygen that this planet consumes comes from one microorganism. And that is a kind of algae. Algae have chlorophyll. So effectively, they're a microscopic kind of plant life, if you want to look at it that way. Our planet is 85% covered in water. Most of the planet's oxygen comes from algae. We probably, because we're land-based here, maybe there's a fair amount more here that comes from green grass, trees. We used to have green here. From the chlorophyll of our plants, our environment. I'm not going to go crazy about evolution. That's not my area of expertise. We'll talk about how this is GMO, genetically modified organism. That's all about bacteria. We couldn't do that if it weren't for bacteria. You couldn't. We couldn't DNA test. We couldn't find out crime scene things, the forensic aspects. That's all from bacteria. All that. And so there's a lot that we'll go into the disease aspect. We're going to look a little bit at the differentiation. And this is the problem. We have agents that are acellular, which means by definition, they're not living. It's a whole different ballgame that's there. And we'll look at the size and all that type of stuff. Wow. So let's see if I'm trying. It's funny. I tend to do these opening lectures somewhat extemporaneously, meaning I'm not really. I just kind of go with it. I remember what I did in the other class. So it's not exactly the same. When we get more into chapter and following PowerPoints, it'll be different. So, voila. Bacteria and archaea. You're a little bit different. I mean, I'm going to try to give you I better pop up. So maybe you can see it a little bit better if I have to use the board. Okay. Is that okay? So, I don't want to write over there the yellow. If the people who are here with me previously would occasionally laugh and smile, it will induce the other ones. Oh. I need help. Living things. What do we have? DNA and RNA. Gotta have that. You're a living thing. Gotta be made out of cells. That's cell theory. Right? You gotta have a boundary of some type. Semi-permeable membranes. Stuff can move in and out. That's sort of a subset of the boundary. You have to be able to respond to changes in the environment, stimuli, stuff going on outside. Got to replicate. And you have to be able to do energy transformations, metabolism. And that's the beauty of utilizing bacteria. They're very similar to us. They're much more simplistic, but they're like us. Rather than being multicellular like us, so bacteria, if you want to look at the distinction, they have DNA and they have RNA. Okay, but they only have a single cell. They all have a cell wall, and that's really significant. Okay? Down the road, you're going to become very acquainted with this chemical substance called peptido glycan, glycan, because their cell walls all have it all. Okay? So a cell wall with peptidoglycan, that's there. They have semi-permeable membranes. All that other stuff. They respond. They do energy transformations like we do. They ingest organic, most of them that we're going to become acquainted with, like us. We ingest organic materials. We break them down. We use some for energy. We use some for building blocks. We excrete the rest. Okay. That's energy transformation, basic metabolism and excretion. So they're very similar. The beauty of why bacteria are so helpful for us is when we try a new medicine, we use it on bacteria first. Okay? Because we have a vast population. In the lab, you're going to put things on culture plates. In a day, 24 hours, you're going to take one drop of bacteria, and in 24 hours, you're going to have millions of bacteria. Every 20 minutes, they divide in two. We're going to get into the gross. It's called exponential growth. It's like if you had a penny and you doubled it every day, you can figure out how long it would be before you have $1 million. 1 cent, 2 cents, what? Do the math. It's called 2 to the n. Bacteria every 20 minutes. Every hour, they replicate 3 times. So you start with 1, you have 2, you have 4. In an hour, you got 8. Gone. So you get vast numbers, and we can apply chemicals or test antibiotics on them and see if it works. And if it doesn't, if it makes the bacteria, if it hurts them, they're not going to sue you. Okay, so you're a living thing that we have. Look, there's no people for the ethical treatment of bacteria. There should be. Let me start that. But in the old days, they did animal testing, and they did a lot of people testing. They don't do people testing until it kind of clears those kinds of hurdles. And indeed, just to show you the importance, bacteria unfortunately mutate because they're genetically unstable. Their DNA is different than our DNA, and we'll get into the structural difference. It's not your mom and dad's DNA, and it's not your DNA. And there's a test that we do. It's called the Ames test, and we'll get to that when we talk about genetics. Any medicine, any antibiotic, anything that they would give a person, before they are allowed to, before we can even do human testing, before they would even start the whole process of the testing, which is very, very expensive, they do the Ames test. They actually mix some of that, whatever medicine, chemical, antibiotic, and they give it to a bacteria. It's actually a kind of salmonella. And they see if it will mutate that salmonella in a specific way. We'll go over the specifics when we do the genetics chapter. If it mutates the salmonella in this specific way, it will not be used on us. It will not be given to people because it's very, very likely to cause cancer. So we test it on the bacteria first to make sure we're not giving something that's more likely to cause cancer than that. And so we're utilizing this genetic instability of bacteria, just for instance. So we have those types of things. So bacteria differ. How do they differ? Our DNA is what? It's in pairs, right? Linear DNA in pairs. That 23andMe stuff, XX, XY, right? Ringing a bell. We had this stuff in A&P. You had that stuff in high school. Okay, so you have DNA that's in pairs. It's linear. We'll get to the beauty of it. Pairs, you get dominant and recessive. If there's damage on one of those pairs, maybe you don't necessarily create a problem. We'll get to that when we talk about the genetic aspect. Our RNA in our cells, so let's say this is eukaryote. Our cells, eukaryotic cells. The RNA is classified as ADS. Where does that come from? That's the domain theory. The domain theory came out in 1978 based on molecular biology, which we can start to chop up DNA and RNA, and it looked at DNA sequences, and it looked at RNA, ribosomal RNA density. And so bacteria, the density of the RNA, just go with it. You're going to learn more about it. Is 70S. You'll get to it. It's a fancy name. I just use the S. They're called Svedberg units. They spin it in a centrifuge and they get the DNA. Our DNA is ADS. There's a difference. Our RNA is ADS. If I said DNA, I misspoke. Their RNA is 70S. The DNA linear in pairs. Bacterial DNA is a big circle. The DNA is the same. Good old double-stranded DNA, you know, Watson-Crick model, you know, spiral staircase, more today than yesterday. That's a song by a group back in the 60s called the Spiral Staircase, Icon Crest. How many musical references? I was listening to a group that had their first, this morning had their first hit in 1962. They took their name from a bowling alley in Union, New Jersey that I used to bowl at, called the Four Seasons. No Four Seasons fans on oldies channels? You have no idea what I'm talking about. You do. God bless you. My day is complete. I used to sing some of their songs to my soon-to-be 40-year-old daughter. Don't tell her I do that. So there's a difference that's there, but there's a lot of similarities that are there. Archaea are very much like bacteria. The difference with archaea, think archaeology, they're older, but that's not the big difference, is where they live. They live in extreme environments. Polar ice caps, bottom of the ocean, volcanic caldera. They're not relevant to our course per se. They are being studied by microbiologists who go to these extreme areas to understand they may have impact on us moving forward in our world. I've read some stuff about energy transformations. A lot of the kind of techno thriller authors that I kind of like, who are kind of jumping ahead of the learning curve of, you know, thinking that, you know, they've heard about certain kinds of these exotic, they produce energy that we might be able to harvest somewhere in the Arctic. So I like that kind of stuff. And I mean, and a lot of that is, there's a lot of real life stuff because a lot of them do research. I mean, I just finished the most recent, you know, Dan Brown is the author. I just finished his most recent book. He only comes with a book about every several years. A lot of that is very relevant to what we're studying, you know, to consciousness and a lot of the neurology stuff that we did in AP1. He talked endlessly about GABA. You guys who have me remember gamma-aminobutyric acid, that usually inhibitory neurotransmitter. That book was all about that. Very, very interesting. So, I mean, they do their research and their homework. So bacteria and archaea are very similar, except where they live. And what's interesting about that, the archaea don't have, even though they have a cell wall, it is not that peptidoglycan. So for instance, what can we get out of this so far? This is the 70S, this is the 80S. This is obviously a difference. When we want to treat an infection with bacteria, that's why we study. Why do we look into all those things, all the anatomy and physiology? Why do we sit there studying that? So because we look on that basis, on that molecular basis, to find out, is there something we can use to help people? Or unfortunately, some people look in the other direction to hurt people. When you think of biological and chemical weapons. This, from a science perspective, so So let's say you had typical infection. Somebody said you had staph infection or strep throat or anything along those lines. What do they usually do? What do they give you? I heard that, antibiotics. Name an antibiotic. Amoxicillin. That's a cousin of penicillin. Absolutely correct. So what's different between those two? What we have here, it works on a significant difference. It so happens the one thing that bacteria have that's unique to bacteria. Unique is not just special or exceptional. It means no other living thing has it. Penicillin doesn't allow the assembly. It interferes and it breaks down peptidoglycan. That's why the worst 3 words I could ever hear when I talk to a patient, are you allergic to any medicine? If they said allergic to penicillin, it's like, that took away my best weapons. Because the big problem with penicillin is it comes from mold are allergies. It has very, very limited side effects because you're attacking something that we don't have in our cells. So let's say you were allergic to penicillin. There's somebody who's allergic to penicillin. What would they get? You can't use the amoxicillin. You can't use regular penicillin. What do they get? Other antibiotics? Anybody take an antibiotic? We're going to take five capsules and it was done for what? What was it called? The word Z-Pack in your vocabulary? Prednisone is not an antibiotic. Prednisone is an anti-inflammatory. We hopefully will learn a lot. We'll cover it. Okay, so the Z-Pack, other ones would be, for instance, a lot of students they take mini doses of either doxycycline or minicin for acne or erythromycin. So typically, if you're allergic to penicillin, the drug of choice is in a family of what are called erythromycins. And so they kind of played with it. So you only had in the Z-Pack, which is called azith. We'll get to all this, don't write it down. It targets the other difference. Here, it attacks 70S ribosomes. It's not as effective. The big problem is side effects. Why? Well, we have 80S, that's true. But inside of our organelles, inside of mitochondria, in all of our cells, we have some 70S ribosomes. You're going to learn that they probably came from our bacterial ancestors that incorporated our cells. The fancy name we're going to learn about called endosymbiotic theory. It's a really important part about microbiology. That's why that's why one of the areas where a lot of antibiotics called side effects. A lot of them target that 70S ribosome, particularly with people who can't tolerate penicillins. That's all coming in our antibiotic discussion. Just for context. Protozoa. So in high school, a lot of people, even me, they did have microscopes back then. Listen, if you were on your game, you would say, you can't. No. God bless you. You're already in trouble. A permanent state for many of you. Now I lost my train of thought. Oh, things in pond water, protozoans. You used to look at the pond water and they'd be spinning around and all that, rotifers. Those are protozoans. They are eukaryotic cells like ours. And the problem with attacking them is they're like our cells. So anti-protozoan medications have some significant problems. The single biggest killer in sub-Saharan Africa, the bulk of the continent of Africa, it's not AIDS, by the way, it's malaria. Malaria is a protozoan. The second most common sexually transmitted disease in this country is a protozoan. It's called Trichomonas vaginalis. Number one, HPV, which is a virus. Number three, herpes, which is a virus. In between, before we get to any other bacteria like chlamydia or gonorrhea, okay, we have protozoa, Trichomonas vaginalis. Hard to treat. Fortunately, there's a very good medication to treat it, but normally we don't have medications that are able to target it that efficiently. Anti-malarial medicines have a few more side effects that are there. Fungi, most common fungal infection, athlete's foot, jockey's, that kind of stuff. Basically affects your skin. Big problem with most of the time. We usually use topical agents. The big problem is they itch, we scratch, and it predisposes, it breaks down a barrier, and we get bacterial infections, what we call secondary bacterial infections. But fungi are cells that are like ours, so when we have a really deep fungal infection, something that gets into your respiratory passageways, it's a lot harder to treat. Fortunately, we have a lot of mechanisms that prevent it unless your immune system is depressed. So here's an important little nugget. In 1981, the paper was released that defined the condition you and I call AIDS, and it pointed the finger accurately to another virus we will learn about called human immunodeficiency virus, or HIV. That paper came from San Francisco because a physician or a physician's group who treated a lot of gay men, the two largest populations per capita, percentage-wise, of gay men back in the 70s, and when the onset, until we identified AIDS in 1981, were in San Francisco and Philadelphia. I was seeing the cases in Philadelphia. I was in school in Philadelphia and a resident in Philadelphia between 73 and 79. Resident in the sense of a medical class. Long before the diagnosis, I'll give you plenty of stories about that. He identified a fungal pneumonia that was very rare, and he had a ton of cases in gay men. It was associated with this virus, which had killed their immune system to the point where they developed a fungal infection that you very, very rarely saw. Called PCP pneumonia. It's a fancy name. And that was the definition of what he called acquired immune deficiency syndrome. And he found the virus was the commonality, both statistically and causatively, in that particular condition. It existed. I was treating AIDS patients long before. I was having lunch with people with this.
Without
Absolutely. Excellent question. It's all about that. We see a lot of those things. Another one. I was telling the other classes a little time. I wanted to cover everything I thought I'd cover with them. We have a couple of minutes. Very common. Most common fungal infection of the athlete's foot, vaginal yeast infections. It's normally there for a lot of us. We typically see that associated with a variety of things. Relatively easy to treat. I was on immunosuppressants a couple of years ago when I was treating my cancer, which thankfully is, for now, bye-bye. Okay, and I stopped taking them because I was taking them because I had an arthritic condition that developed. And I was getting frequent bouts of thrush, which is the same thing, a fungal infection called Candida albicans, same thing that causes vaginal yeast infection, because we have a fair amount, about a third of us, half of us have it in our mouth. And I was getting it frequently. Fortunately, it was easy. Just had lozenges with antifungals like 5 times. You got a sore throat and burning and that you can only imagine. These are worms. Forgive me, I'm not putting the worms in. When the worms start, they're microscopic. As they get bigger, they become macroscopic. I don't know enough about worms to teach you. Here's what I tell every class: if you have a pet, ask your vet. They are the pros. We don't have a lot of problems with this because we have a pristine water supply. When people get worms and things like that, it usually has to do with a compromised water supply. We do not see it, other than when there's a natural disaster. But in a lot of, unfortunately, war-torn countries, natural disasters, you see tons of this. No problem. Algae, I commented on before. We'll get more into it. Bad algae, red tide. For instance, that could be a potentially poisonous algae to you. By and large, microorganisms are your buddies, but not viruses. Viruses are not living. They don't even have DNA and RNA. They have either DNA or RNA, never both. They don't have any kind of organelles. They have protein shell and maybe a little bit else, period. They're elegantly simple. That's the whole thing with all of these things. The simplicity of the designs of these microbes, there's not a lot of moving parts. You know what they do really well? They reproduce. That's the big problem. Why we can study them, they reproduce quickly. Why they hurt us, they reproduce quickly. I mean, it's just like with my wife's influenza. Well, why didn't we take her? After about 2 days, antivirals for influenza don't work. They're very inefficient. Tamiflu and things like that. You got to get it in the first couple of days. She wasn't moving. They're mobile. So viruses, DNA or RNA, never both, with protein shell, not a cell, not living. Prions, protein infectious particles. Proteins only. They're misfolded and they bang into other proteins and cause them to misfold. Most famous disease, and they may all be the same, mad cow. So we have a unit on that as well. That doesn't come until our second test unit. We really get into viruses and all this stuff. Lastly, you'll be happy to know that within a couple of minutes. You can look at the size charts. They're small. I mean, if that's a eukaryotic cell, okay, some of these big eukaryotic cells, okay, like let's say this was a blood cell like that. Viruses, here's what E. coli would be. Here's what staph or strep would be like. Comparatively speaking, reproduce rapidly. We use them in the lab. They're easier going to do it yourselves. You have to have the microscope. That's what lab is about. And we'll stop there. Thank you all very much. I think I got most of the stuff in that I covered with the other group. No guarantees. I'll try to be more organized. GFL.